In this video of the cell cycle, you're going to learn the role that the cell cycle plays in a variety of activities. It's not as easy to develop and use a model to explain the role of cellular reproduction unless you understand where some of the components come from. What happens while they're developing, how it differentiates, how it divides, and then how it regulates itself, and then of course when the regulation goes awry. To better understand this duplication, let's do some DNA vocabulary. A chromosome is one long continuous thread of DNA that consists of thousands of genes, and that's usually just regulatory information. A gene is a specific section of that DNA that contains instructions to make a protein. Each human body cell has 46 chromosomes in it, and that is how the DNA is organized in humans. Half of your chromosomes, which is 23 of them, came from a biological mother, and the other half came from your biological father. That's why these things occur in pairs. And so your DNA is organized into 46 chromosomes, or 23 pairs. And they have thousands of genes on them that provide instructions for making proteins, which ultimately create you. A chromatid is going to be one half of the duplicated chromosome. A sister chromatid means that there's two identical chromatids. So if I have a chromatid right here, I have an identical chromatid right there. All together, I have a chromosome. And the center part that kind of ties them together is called the centromere. It's a region of very, very condensed chromosomes that look kind of pinched together. So inside of your cells, for instance, in one cell, you have unduplicated chromosomes. Here we have, we'll just say this is chromosome number 1 out of your 23 pairs. It has half that came from your mom and half that came from your dad. So this is a chromosome. These two items right here are called homologous chromosomes. They're the same, they're just a duplicated pair. This is how it exists in your body. When it gets ready to divide, it's going to split up into two identical parts. So this is the original, and this is a duplicated. Here I have an original, and here I have a duplicated. So one chromosome has turned into two chromosomes. This is all during S phase of interphase. The reason the cell is doing this, and every cell does this, every ear cell, every chin cell, is because it's getting ready to divide. And when it divides, it's going to need an exact copy of everything for the new cell. The part here right at the center of all of these unduplicated and duplicated chromosomes is called the centromere. Now that we've completed interphase, we're ready to divide. So we have completed growth, and then duplication, and then growth again. And so we are as large as we're going to be, and we're going to start dividing. Mitosis is the division process. It's where one cell becomes two identical cells. We usually call this first cell the parent cell, and the two identical cells that follow are the daughter cells. So this would be the parent cell, and it creates these two daughter cells. In mitosis, we go through the process of what I call PMAT. That is prophase, metaphase, anaphase, and telophase. These are the steps to mitosis. The last part of the cell cycle is cytokinesis. In prophase, the chromosomes are very visible. They're visible as sister chromatids. Remember, that's 2 copies of the exact same chromosome, and which are represented as X's here in the diagram. And the nuclear membrane disappears. So we no longer have a nuclear membrane around where the DNA is. It just goes away. And then these spindle fibers come out, and here come the centrioles. Like I said before, they looked kind of like a sun. So here come the centrioles with attached spindle fibers, and these spindle fibers are going to grab onto, what do you think? Yes, this little center part, the centromere. The spindle fibers connect to the centromere of each sister chromatid. They line up, hence meta, middle. They line up in the middle of the actual cell. You start to see the fibers attach right here, and the centrioles move to opposite ends of the cell. In anaphase, your chromatids are going to move to the opposite ends of the cell. So these spindle fibers contract and pull apart the sister chromatids, taking half of the DNA with them. So sister chromatids, they separate, pulling away from each other and becoming individual chromosomes. In telophase, the chromosomes condense and start to look like chromatid again. Remember, chromatin is like a little thread-like material. When the cell's just hanging out, you don't see the actual chromosomes in them. It's just relaxed. They're not condensed. They're thread-like little rods. And the nuclear membrane starts to form around again because we want to keep these guys separated. Through all that hard work, we don't want anybody getting out or into the other cell. And so the nuclear membrane forms around the chromosomes at each pole, and the spindle fibers break down, and we begin cytokinesis. Cytokinesis is the division of the cytoplasm. The cell is going to take all of its stuff that it made from G1 and G2 and split it, divide it amongst both cells. Now, when this happens in plant cells, this will form what we call a cellular plate. And that's the division between the nuclei, and it's very gradual, and it develops into a cell membrane. Kind of starts to form midway, and it happens very gradually in plant cells. In animal cells, it forms through what we call a cleavage furrow. So cleavage, the dip right here, it pinches off, putting the cell into 2 equal parts. Now every cell does this, it divides, and it's usually at a different rate. It really depends on what type of cell it is and how often it needs to divide. So for example, your internal intestinal cells, they divide about every 5 days. Skin cells, they divide about every 2 weeks. Red blood cells take a lot longer, about every 4 months. And liver cells, you could have the same liver cells, their cellular division is very slow. It could take a year to duplicate them. But why do your body's cells need to divide like in the first place? Besides the most often, or the most obvious reason, which is to take you from that zygote to the beautiful human that you are, cells need to repair themselves. They just can't get bigger to grow. They have to stay small. They have to maintain that high surface area to volume ratio. That allows them to move stuff in and out and maintain homeostasis more easily if they stay smaller. So this is the most efficient, energy-wise way that allows substances to move in and out of the cell's body easily and maintain homeostasis.